In this chapter, we are going to discuss about Mendel's laws of inheritance and Mendel's experiments. Mendel's experiments are carried out on pea plants which are included our syllabus. The pea plants are selected and it has special reasons which are very essential and they are discussed in last lecture. These pea plants are not only suitable but they have the contrasting characters, contrasting traits, and these traits are seen in this diagram. You can observe the 7 traits, 7 characters one by one. These characters are related with seeds, flowers, pods and stem. About the seeds, it is about a shape of seed as well as color of cotyledons. Then the cotyledons are also having two, yellow and green shape, has round and wrinkled. Then the characters about the flowers, white flowers and violet flowers. Next one is about pods, full shaped pods and constricted pods. Next one is color of pod, yellow and green. Then the last one is a stem, that is the flowering on the stem. One type is axile, another one is terminal, and another that is length or height of plant, long and short. It is also called as tall and dwarf. These 7 characters are contrasting with each other. Hence, these are helpful in experiment as well as helpful in a recording of the results. These experiments gives same results. Mendel conducted experiments to determine the laws of inheritance. These experiments were divided into groups. Out of that, the monohybrid cross and dihybrid cross are included in our syllabus. The experiments gives some conclusions. These conclusions are as the genetic makeup of plant is known as genotype. On the contrary, the physical appearance of plant is known as phenotype. The genes are transferred from the parents to offsprings is in pairs known as alleles. During the gametogenesis, means gamete formation, when chromosomes are halved, there is a 50% chance of one of the 2 alleles to fuse with other parent. When the alleles are same, they are known as homozygous alleles, and when the alleles are different, they are known as heterozygous alleles. These are the some terminologies which are utilized in experiment. The monohybrid cross means the experiment in which Mendel took a 2 pea plants of opposite traits. For example, one plant is short, another plant is tall, and crossed them. He found the first generation of offsprings were tall and called it as a first F1 progeny or first filial generation. Then he crossed F1 progeny and obtained both tall and short plants in a ratio 3:1. To know more about this experiment, visit the monohybrid cross inheritance of one gene. And from this experiment, from this cross, he postulate the laws of segregation and law of dominance. This experiment, in which tall plant and dwarf plant is selected, and it can be schematically represented as shown in this diagram. The cross, which consists of two contrasting characters, that is tall plant and dwarf plant. These plants, tall or dwarf, means this is a phenotype, and their genotype is mentioned in colorful letters below that, capital T and small t. They produces gametes which include a single set of allele, means the tall plant produce gamete which has a one T, single T, and dwarf plant is also produce gamete which has a small t. And when its first phylyl generation is developed, it has such a combination, that is capital T and small t. The two different genes are present, hence called as heterozygous tall. That is, these all the first phylyl generation appears tall, but genotypically they are having the both type of alleles. Then Mendel cross these F1 plants selfing, and after selfing the free pollination is allowed and both type of gametes are freely combined, and their result, which is seen in a next, that is F2, second phylyl generation, is shown in diagram. The technically a suitable method is checkerboard. In this board you can observe the male plant gametes, female plant gametes, and free combination, possible combinations, which produce 4 types of offsprings, and these offsprings shows a phenotypically in two groups, that is tall as well as short, but this tall has a more in number, near about 75%, and dwarf are less in number, that is nearly 25%. This ratio is 3:1. It is called as phenotypic ratio or standard monohybrid ratio. Genotypically this ratio is 1:2:1, means the digit 3 for tallness is having two types, homozygous as well as heterozygous. This cross shows a one example, but similar results are found in all 7 characters of pea plant, and hence all these similar results are called as the laws or principles. We can see the another example in which another character is considered as a yellow and green. This example you can observe in this diagram. If crossed yellow and green, and F1 all gets the heterozygous genotype and appearance is yellow, but when selfing is there, both parents, which are heterozygous, produce two types of gametes, and Hence it produces similar results, that is 3:1 phenotypic ratio and 1:2 2:1 genotypic ratio. And this is true for all the 7 characters of pea plant. Hence Mendel postulate these results as a laws. First law of Mendel, that is law of unit characters. Mendel postulate these laws with a different terminologies. In this law, each character is controlled by distinct units. These units are called as factors, which occur in a pair, and if the pairs are heterozygous, one will always dominate to the other. In modern science, the units or factors are called as genes. These units means Mendel says that there is something which carries the information from parents to offsprings, and this something is called as units, and unit is also called as factors. And we, after the microscopic study, now we can call it as a genes. The next result of this monohybrid cross is law of dominance. According to the law of dominance, the hybrid offsprings will only inherit the dominant trait in the phenotype, and the alleles that suppressed are called as recessive trait, while the alleles that determine the trait are known as dominant or dormant traits. This is a law of dominance, and it is true for all the 7 characters of pea plant. Then the law of segregation. This is also a one result of observed in this monohybrid cross. According to law of this segregation, it states that during the production of gametes, 2 copies of each hereditary factor segregate so that offsprings acquire 1 factor from each parent. In other words, alleles or the alternative forms of gene pairs segregate during the formation of gametes and reunite randomly during the fertilization. Means the different alleles are getting segregated during gamete formation. This is a law of segregation, and we see in this monohybrid cross, that is gametes get separated. In the next experiments, the Mendel select the parents having 2 different characters, and then experiment is called as dihybrid cross. In dihybrid cross experiment, Mendel considered 2 traits, each having 2 alleles. He crossed wrinkled green seed and round yellow seeds, and observed that all the first generation progeny were round yellow, that is similar to dominant parent. This meant that dominant traits were the round shape and yellow color. This experiment can be shown in diagrammatic representation as follows. In this diagram, we can observe first parent which has a yellow round seeds, that is dominant character, hence written as a capital letters, and another parent is having recessive, that is the green and wrinkled seeds. In this cross, F1 generation, which is heterozygous. In diagram, F1 is written as a capital Y, capital R, small r. In this generation, these gametes as well as these alleles of two different traits are combined. They are present throughout the generation, and then when they became a mature, they form gametes. At that time, both are showing a independent combination during gamete formation, which is observed in next chart. This parent, when selfing is done, that is both F1 plants are crossed, both are having a genotype capital Y, capital R, small r, and such a plants which having a two different characters in heterozygous condition, they produces a four types of gamete, and due to such a four types of gametes, both plants produces the 16 possible combinations. These 16 combinations are called as F2 generation, and this F2 generation shows a result phenotypic ratio 9:3:3:1. In this example, we can see the characters which are assorted, which are separated independently. Same example can be studied in different way, can be observed in this diagram or understand in this diagram. You can see the results, same 9:3:3:1, and this diagram, which is showing after selfing of F1 progeny and obtain four different traits: wrinkled yellow, round yellow, wrinkled green seeds, and round green, in the ratio 9:3:3:1. In this ratio, we can observe the next result, that is law of independent assortment. The law of independent assortment states that a pair of trait segregates independently of another pair during gamete formation, as the individual heredity factors assort independently. Different traits get equal opportunity to occur together. Means the opportunity of mixing of genes, possible combination, is independent. The traits also get segregated, separated. They show the number is different, that is 9, 3, that is dominant characters shows more combinations. One character shows the only one dominant and one recessive. It is in 3 number, but combination is independent. Hence, the law of independent assortment is postulated on this basis of experiment dihybrid cross. In this way, the Mendelian principles which are done with on the basis of the experiments carried out on the pea plant. In next lecture, we see the different failures of these laws.